Okay. People are real sick in the ICU, and they are unstable and moving around. We observe them with a lot of uncertainty. We don't know exactly what's going on. We want to do our very best to understand what's going on so that we can manage oftentimes the simultaneous and confounding issues of fluid balance, which can range anywhere from, severe venous overload to hemorrhagic hypovolemia. And at the same time, their renal function is changing, and the excretion of their drugs is changing because of this. And we need to keep up with all of this. Now, a lot of people in the ICU have large apparent volumes of distribution of the drug in them, in many different drugs, perhaps because of changes in capillary permeability as they are ill. And recently, the issue has been called that of augmented renal clearance, and Jason Roberts in Australia is a big proponent of this, and has done a lot to promote the idea and the awareness of augmented renal clearance in patients in the ICU. And what we might do is to examine that and see if that's what we really want to know, and what we can do to take the best care possible of these acutely ill and highly unstable patients. So here again is our lab. There's the old geezer, and there's Mike Neely, our new director, who is so good, and I'm just so glad that we've had such a good relationship for the last 10 years, 12 years. And again, Alan Shumitsky is professor of mathematics. David Bayard is a mathematician who works in the Jet Propulsion Laboratory in the guidance and control section. Mike Van Gilder is our main scientific programmer. And Ida Bustad is the queen and the empress who runs the entire operation. It would fall apart without her. And so here we go. And come on now. Let's ask first, why did God make kidneys? We are all so caught up with looking at creatinine clearance and the idea of clearance and GFR. And why did God make kidneys? Did he give us kidneys so that he could make urine? I don't think so. I think he gave us kidneys so that we could get rid of waste products from our body. And so the idea of looking at glomerular filtration rate is a useful one. But I think it would be at least as useful to ask ourselves, for example, what is the half-time of creatinine in a particular patient? And how rapidly is the patient getting rid of whatever toxic substances we want to talk about or discuss? And so we might come to the idea, and this is heresy. This is heresy. Everybody knows that I'm crazy. And everybody else knows that clearance is often felt to be the most physiologic parameter in pharmacokinetics. Well, let's forget pharmacokinetics. Let's talk about how we describe the behavior of systems, whether they're pharmacokinetic systems or radioactive decay, or the equations that describe the behavior of satellites or aircraft. And for any PK system, we really don't need clearance. Clearance commingles the idea of the volume of distribution and the rate constant, because it's the product of those two things. When you multiply them together, you have a number, but you lose the information of the relative contribution of volume of distribution and drug elimination rate when you do that. So when we have any pharmacokinetic or any system like radioactive decay. The equations that describe the systems can be described as the amounts of the drug in various compartments. And we can have rate constants describing the rate of movement between compartments. The inputs are the doses, and the outputs are the observations, the amounts in various compartments divided by the apparent volume of distribution of that compartment, or other responses that do with effect, for example. Now, if we go and look at the change in amount in a compartment per change in time, it's usually minus, because it's decreasing, let's say, from elimination, the rate constant times the amount, where A is the amount, KE is the rate constant. Now let's let the volume be V, so that a serum concentration is the amount divided by the volume. Now clearance, we know, is the rate constant times the volume, and of course we can rearrange that to say that the rate constant is clearance over volume, as people say, and of course the other way around, the volume is clearance over the elimination rate constant. Volume has units of liters, whatever some volume unit is. The rate constant has units of reciprocal hours. So clearance, when you multiply the two, has units of liters per hour. And it's only the rate at which the volume is cleared is seen, and the true information about the rate of drug movement or of the volume itself is obscured by commingling them. And that's the problem with clearance. But when I say this to people, they all know I'm nuts. So, is it more useful to describe behavior as volume and clearance or volume and rate constant? Again, clearance commingles. The rate constant, or the half-time, is the parameter that gives direct information about the rate of movement of the drug from one compartment to another or out of the body. And for unstable ICU patients who often have problems with fluid balance and changing renal function, it's more useful, I would suggest, to separate volume and rate constant as two separate issues so that you can see better what goes on. At the Cleveland Clinic, back in the 1980s, they've used our software for many years, and Marcus Haug and Peter Slug there got the idea that here is a patient, and they didn't do like I told them to. They instead of fitting all the data, they would fit this cluster when it came back, and then they would fit this cluster when it came back. And then they'd say, look, this guy's volume of distribution is getting smaller. And they finally got the idea that it was associated with recovery. And so they said this patient has VD collapse, and that means he's going to get well. So they were seeing that version of the phenomenon. Now we see Jason Roberts and many people talking about augmented renal clearance in acutely ill patients in the ICU. And so that's the other side of the coin. People have often big volumes of distribution, and then when they get better, it gets smaller. And so that's the idea of augmented renal clearance, which I think we do better by separating it. So for elimination, we can say that renal elimination is the renal rate constant, which is often related to creatinine clearance, so that we can describe it as a slope relationship or some number times whatever the patient's creatinine clearance is, so that we can relate elimination to creatinine clearance. The non-renal elimination is another number for rate constant, and so the total elimination is the total of those two. Now, people in the ICU often get dialyzed, or they get put on ECMO or renal replacement therapy. How do we include that in the analysis of the drug behavior in them and of their fluid balance? And this is an important thing, and it's very simply done. Here's a patient who had sepsis a number of years ago, and his baseline creatinine clearance was 3. There we see it here. and he got a dose of 100 milligrams of gentamicin back then. But this is the important thing. At this time, he began to be placed on dialysis. And so what you do then is you give him a dose of 0, and you set the estimated creatinine clearance at some other number that you think he might have. Here I set it at 50, presuming that an ordinary episode of hemodialysis would contribute maybe an excretion equivalent to about 50 units of creatinine clearance. So there it is. Now when he comes off, he gets a dose of 0, and his creatinine clearance gets set back to its original baseline figure. And in that way, you can include this process in the ongoing analysis of the behavior of the drug in him. And here we see he got another one here, another one here, and it's an easy way to include the process of dialysis or ECMO or renal replacement therapy in the analysis of his ongoing therapeutic problems. Here's a patient, this patient, and here we see, this is just the analysis with his population model before fitting it to his serum concentration data. And here, these gouges like this are the episodes of dialysis. And this took place just as our intensive care unit, back in the early 1980s, began to have 24-hour pharmacist coverage. And now we see an interesting thing, because everybody had the feeling of teamwork. And yeah, that's great to have you guys aboard. And so everybody wanted to be part of the team. And they were saying, you know, the serum levels are less than the population model would predict. But they didn't really say that. They didn't want to offend anybody by saying, you know, you got to give bigger doses. So they were saying, well, he's got a large volume of distribution. And the physicians, who had never had any pharmacokinetic training, because no physician in any medical school in the world that I know of gives it in any clinically meaningful way. So the physicians didn't know what that meant, and they didn't want to ask. And so the message did not get delivered that this patient needed bigger doses. Here, when you fit the data, the model to his levels, we see the lower serum levels. And back up here, this one, you see, is a peak of about 8.5, but more likely, it's only about 6. So that's a big difference. The pharmacists were giving the gentamicin in the ICU. Now, he got well enough to leave the ICU, and he went out to a general medical floor, which did not have pharmacist coverage. And so his gentamicin orders did not get renewed or continued. And so we see here that he went for about a week with no gentamicin. At that point, a new crew of interns and residents came on, found the problem, began to give him doses, and they asked us to come and see the patient. And we did. And this is the situation as we found it. But the patient didn't get better, and he died out here because the infection was not able to be controlled again. So that was a real sad thing. And the lessons from this can be that both physicians and pharmacists need a common knowledge base in order to communicate. And we need to have physicians have meaningful training in pharmacokinetics. And most MDs don't know what a volume of distribution is or a rate constant. Oh my God. And you can include the data of these procedures this way, as we have just seen. And it's also useful, oftentimes, to dose before rather than after dialysis. Because if you give a dose of an aminoglycoside, for example, before, a couple hours before, then the dialysis itself produces a situation in which renal function appears to be more normal. And you have a slightly more normal relationship between peak, trough, and AUC than when you give it after dialysis. So I think in general it's useful to give it a couple of hours before dialysis for aminoglycosides at any rate. Now here's another patient who started out here, not in the ICU, and this was a lady with a pyelonephritis. And she had a peak of 4 point something here, 4.6, I think, and a trough of 0.4. And she was doing well clinically, and everybody was pretty happy. And then all of a sudden came the surprise that she relapsed and went into septic shock over here. Why did this happen? When they went and looked at her data, they see that she missed a dose here, and here she went for a whole, more than a day, and here for another day without any gentamicin. And so that's probably why she got so septic. Now look at the very poor relationship between the predictions, estimates of the serum levels and the actual measured levels. It's a terrible fit. And this is the problem of all current clinical software when it comes to managing acutely ill, unstable patients. What we did at the time was to break this data up and look at her before she got so sick and then after. Over here, her volume of distribution was really pretty normal, 0.25 liters per kilo. But here, it was 0.55 liters per kilo. Big difference. Now, what we want to be able to do is to track the behavior of the drug in the patient as precisely as we can, especially in these unstable patients with rapidly changing volumes of distribution and rate constants. Here we see the bad prediction. And here now we go to the aerospace industry. And the main purpose of a tracking system for air traffic control or for air defense is the estimation of target trajectories, just like serum concentrations, target trajectories in the controlled area and their prediction into the near future. So this is how you shoot down enemy fighters taking evasive action. And it's the same thing. You want to track the behavior of the serum concentrations. You're going to need more data than usual with these rapidly changing patients. And you're going to need also software that tracks the behavior of the drug, just like the behavior of the plane, and lets the parameter values change with each new data point as it comes in. And here's the improved fit that we get with this software, which I think is unique to our software and lets you deal with this problem in this manner with these unstable patients. And here we see the better predicted found relationship. And then here, actually in the software, there's a little arrow here. And you can take that arrow with your mouse and click it here, and you will see either the population model, in this case, or the Bayesian posterior after these levels. And then you can see it change after each new data point as you move the arrow through here in time. And in this way, you can track the changing volume distribution or the rate constant. And you see that they change independently of each other. And so you need to know them separately this way, and separating them this way, rather than having them commingled as clearance and volume, gives us, I think, the most useful and clear clinical information about the behavior of the drug in the patient and about the behavior of the fluid balance in this patient. So that's what we like to do, and here we see the changing PK parameters over time this way with this software. When we go back and look at our first patient, now with the IMM, again we see the better fit, and we also see the dialysis, and here we see the fluid rebound as the drug from the peripheral compartment comes back into the serum levels, into the serum compartment after dialysis. And so here we see the changing volume and rate constant in this particular patient. Now this data is old, and nobody back then had the idea of putting this all together this way, because we didn't have the software back then. We were just beginning to get the idea for it. But if we put it all together then, we can get samples of serum creatinine and the drug concentration at the start and end of dialysis or renal replacement therapy or ECMO, and we can get also peaks and troughs or peaks and D-optimal or MM-optimal designs, and we can use the clinical software tools with the non-parametric models, which we need, and non-parametric Bayesian analysis of individual patient data, especially for the IMM clinical software for very unstable patients, like we see here. And this is what I would suggest that we consider using to do a better job of taking care of these unstable ICU patients. Now, when it comes to evaluating renal function, everybody uses either Cockcroft-Gault or the MDRD methods, and the renal people like that idea because it's simple and easy, but it doesn't work. And the problem is that you have to assume that the serum creatinine is stable, and in these people it is not. Let's look at this idea now. We know that clearance is the urinary concentration times the urinary volume divided by the plasma concentration. We can rearrange this to say that PC equals UV. So now if we don't have a 24-hour or whatever urine specimen, we know that PC is equal to it. And here's the article that we like the best for this. And so if a person has a change, has a decrease in creatinine clearance at this time, the serum creatinine will rise and eventually plateau to a new steady state. But when will this take place? It'll take place maybe in 2 or 3 days when renal function is still pretty good. But it may take as long as at least a week when renal function is real bad. And that's the problem. Anytime that you get a single serum creatinine along here, you're not perceiving the full response that change in renal function will produce. And so your estimate of the patient's change in renal function is always too little and too late after the event. If you use a pair of serum creatinines, so that let's say you get one here and one here, you have a much better idea of the rate of change of serum creatinine, and you can calculate the creatinine clearance that produces that in a patient of that stated age, sex, height, and weight. And that's the way we do it. And also, what are the errors in the classical gold standard of a creatinine clearance that's done in a research setting on a metabolic ward with a 24-hour urine? I think that we probably are the only people who've looked at that. But what people tell us, what did I do? Can you still hear me? No. I did something dumb. There we go. Thank you. What you have is that the coefficient of variation, now we shouldn't be using coefficients of variation, but it's about 5%, like a standard deviation. And of a urine, it's about 8%. And then if you can collect a 24-hour urine with a 5% error, the way those errors will propagate is you square each of them to get the variance, add them up, and take the square root. And so you get 114, and the square root of that is about 11. So the gold standard creatinine clearance has a coefficient of variation of about 11%, 95% confidence limits of 22%. So if we go back now and look at what we got, that's the scatter of this relationship between our estimates of creatinine clearance in patients in our transplant unit and the actual measured creatinine clearance on the books for those patients. The scatter is the error in the estimate of the gold standard. And so we see now that this way we are able, without a urine specimen, in unstable patients, to get an estimate of creatinine clearance that is essentially of equal precision as the classical research 24-hour urine specimen determination of it. So that's a big help, we think. So now when we optimize our protocols, again, don't just do TDM to get a level to see what you've done. Plan it in advance to optimize it. Make it happen optimally so that you can learn most rapidly how to learn about the patient while you're having to take care of them at the same time. So don't wait to get steady state. There's no reason ever anymore to wait for steady state before you get a serum level. That was back from the days of Sachok-Zasky when you did linear regression on the logs of the levels, and that's the only situation in which that now obsolete method would work at all. So there's no reason to wait for a steady state. Just start in with the very first dose and use either D-optimal design or MM-optimal design so that you learn most rapidly. Maybe get a peak and then a sample when you think they're about a third of the peak, or a peak and a trough. And the TDM community can do much better in this area. And you get better care, shorter hospital stays, shorter cost per admission. Now here, for example, suppose that the volume of distribution would change by, let's say, 10%. At what time in this format of giving the dose, at what time would the change in the serum levels be the greatest? It would be up here at the peak. And that's the basic reason for getting a peak, not a half an hour later, but a real peak out of the opposite arm at the end of the IV to learn best what the volume of distribution is in the patient. And then once the drug is in, we see that if you get a sample when the sample is 36% of the original peak, that will show you, that will be the point at which the change in the rate constant for elimination gives you the biggest change in the serum concentration. So these times are the times when the serum concentrations are maximally sensitive to minor changes in the pharmacokinetic parameter values. And that's why they're useful. Now, when we're making PK models, and this is going to kind of review what we went over yesterday, what is the ideal PK model that we could have? It would be the correct structural model, and somehow we would know exactly what the parameter values were in each subject. And the population model would be the discrete collection of each subject's exactly known parameter values. So it's discrete, not continuous, but discrete. And the usual statistical summaries would lose information from that. Now, the non-parametric approach in which the parameter values are not constrained by the assumptions of normal or log normal distributions, these unconstrained parameter distributions best approach this unattainable ideal. Here's again an example that we saw yesterday from Alan Mallet. And we see that here's one group, here's another, but always you have this diversity. How are you going to manage this diversity best to come up with a dosage regimen that will most precisely achieve the target serum concentration that you want a patient to have? Now, here is a nice paper by Michael Neely, and this is a simulation. And here we see that there are some rapid eliminators and some not so rapid eliminators, and pretty much the same volume of distribution. And way out here is one guy who's a way outlier. And when you take that data and you do the non-parametric analysis of it, the gray circles are what you get. If you use parametric methods, this is what you get, these ellipses. And you miss this guy out here, and this is where you think the most likely action is. And so you miss the whole idea of the actual distribution of the parameter values in the population. This is why the non-parametric approach does better and always gets you results that are more likely given the data. And then we develop maximally precise methods with multiple model dosage design. We want to estimate our creatinine clearance from pairs of changing serum creatinines. We want to use D-optimal or MM-optimal TDM protocols. And we're going to use MM or IMM Bayesian adaptive control tools. And we're going to enter the dialysis data as we described it. And so these will help us learn about the patient most rapidly while treating him at the same time. Hospitals need optimal software, and they need a trained dosing service. Mostly this doesn't exist, except in a few places. Natalie Blazak and Lyon sent us this email last April. And in there, this is not the International House of Pancakes. This is the Institut Hematologique Oncologique Pediatrique in Lyon, in France. And with their pediatric bone marrow transplantation center there, and they have the lowest transplantation-related mortality and the highest one-year overall survival in France. And they think that, among other things, that our software, which helps individualize the dosage of these drugs, is useful to them. And so they think that we're pretty good guys. And that's Natalie. Here is Anders Asperg in Oslo. And they have these articles out showing that you can do better using our best dose software with tacrolimus in renal transplant patients in a randomized prospective study, do better than their expert transplant physicians. And also that if you want to include genetic components of that, genotyping helps, but only for the first couple of days. And after that, if you continue getting serum levels, you learn everything that the genetic testing will tell you. And so that reminds us that if you follow the behavior of the drug, you will be acquiring information that reflects the effects of all of these genetic diversities in people, in populations, and also of all the drug interactions that we know of or which we haven't yet discovered. So it really is useful to look at the behavior of the drug. The genetic testing thing is useful for science, but for clinical purposes, it is really most useful clinically to track the behavior of the drug, just like hostile aircraft. So, thank you very much. And any questions?
Any questions?
I have one question. Dr.
All righty,
Jelliffe?
thank you.
Dr. Jelliffe, when I started my work here in the United States, I came to University at Buffalo, New York, to study with Dr. Shantag.
Yeah.
My intention was to specialize in drug monitoring with him. Mm-hmm. Unfortunately, I have to deviate my intention because at that time he had to move his lab and end up not working with him directly. Okay. Unfortunately, I could not do it because by the time I got to University at Buffalo, he had to move his lab from one hospital to another, and that was delaying too long my thesis, so I have to, start with another material. But we made a good, relationship because I stayed with him for a year, and I'm still very interested in drug monitoring. Yeah. And so, what kind of method your lab used to dose those drugs?
Well, to measure the serum concentration?
Yes.
We don't suggest any method for measuring the concentrations. We just want to use the data that people can bring. So we don't favor any particular assay system. We do suggest that it's useful to describe the errors of the assay, as we talked about yesterday, in terms of the reciprocal of the assay variance. so that people can, so that you can separate out the contribution of your assay error from the other contributions of the errors in the clinical environment that are the errors with which the doses are prepared and given, and the errors in recording the times at which the doses are given and things like that. Seems like everybody else just lumps that all together in a single error model, and doesn't seek to ask how much is due to your assay and how much is due to the clinical environment. If you separate that out, and then if you have a large error in your clinical environment, you can know that, and then you can say, hey, maybe we need to fix this or fix that or whatever.
Okay, yeah, that explains why it was so important for you, the assay standard deviation.
Yeah.
Yeah,
Mm-hmm.
okay, got it.
Mm-hmm. Thank you.
Any other questions?
So maybe I'll ask Dr. Satish to give the facility, Dr. York, please.
Well, thank you very much.